Still 22 and 23.
Roger.
25, 27, 26, 25, 26. I mean, right now we're on the lower bound of what your temperature range is, right?
Mm-hmm.
Is that a value to you, or do you want a little bit higher temperature? Maybe.
I think it's okay. I mean, we could stay here all day, I guess.
Yeah, we could.
What I bumped it down a little bit. What's the temperature?
25, 27, 28, 29.
I think
Yeah,
it's just going to be
this
a
is
very
actually the
difficult
most
sample.
stable temperature we've gotten so far, so I think I'm going to take the sample if you think the wrist is locked in and not going to droop.
Yeah, video come a little bit wide, please.
Okay.
That's good. Yeah, it looks all right.
Looks
Okay.
all right?
Frozen. Go ahead.
Yeah, this is pretty good. We're in the upper, we're at about 30 degrees, so I'm going to start the sample now.
Roger.
Okay, it's opening and it's about 34 degrees at the opening. Maybe we can see the coupling. Yeah, we can see the coupling turning on the gas tight. That's encouraging. About 28 degrees right now. 30.
Oh my gosh.
So don't worry.
Okay.
Sometimes when a command is sent down to the IGT, the temperature goes a little crazy. It was registering 273 degrees, which is why Amy said, "Oh my God." It's just a little blip sometimes in the communication when we send a signal down. But right now we're at about 30 degrees, very stable.
Roger.
Cool.
That was good filtering, Amy. You could have said many things.
30 or 32 degrees as we're sampling currently. We're about halfway through the fill.
Roger.
Still holding very steady at 31.5 degrees. Of course, I said that and it dropped to 28.
It can hear you.
28 degrees currently, 29. Another 13 or so seconds for the program to finish, and then it'll start its close.
Roger.
Uh, 32, 33 degrees currently. All right, closing. We should start to see that coupling rotate, and we do. About 30 degrees as it's starting to close. Okay, I'm just going to give it another little bump to make sure it's good and closed. When I say that, I'm just giving the command to close again to make sure that it's closing at full torque.
Roger.
Thanks for the explanation.
Okay, you can go ahead and stow it. Sample completed. Thank you.
Roger. Video, please come wide.
Yep. Nice work. Full wide.
Roger. Thank you.
All right.
So we're going to stow the IGT and stay right here and take some
2
supers.
IGTs in the bucket.
Not literally quite yet.
No, I have confidence you'll get there.
Sorry, Josh.
Uh-oh. I'm just trying to follow you.
So for those of you following on Nautilus Live, that was the second of these isobaric gas tight samples that we were taking at this site. For those of you just wondering where we are, we're at the Secret Cliff hydrothermal vent site.
Oh, yeah, good.
This is part of the Gorda Ridge Formation, about 300 kilometers off the west coast of North America.
The nozzle, see the white paint there? That's where the nozzle
filters
rests
first.
through that little notch.
We're
Okay.
operating.
And then the white there is where the body of the IGT sits.
Yeah. And I'll try doing nozzle first instead.
Yeah,
this
you just
time.
kind of get it in there, and then when you get it close, you just kind of let it go.
We're operating at a depth of about 2.7 kilometers under the ocean.
We can also rock out It can make it easier for you as well.
Yeah, that's a good point. I'll try this out first and
Okay.
then. Oh. Second, let me hold that.
Pushing down on it. Now you gotta pitch up. Pitch up.
This is our second dive as part of the subsea expedition.
Oh, hold on. Yep, okay, you're good.
Oops, sorry. Tried to freeze. Thanks, Josh.
Yesterday we
Yeah,
spent a lot of
swing.
time characterizing the extent of this hydrothermal vent field, and today we're doing, as you can see, some detailed measurements at particular sites. Those orange cones you can see in the background are microbial growth experiments that we placed yesterday. We've just been taking samples of vent fluid, and now we're going to pass vent fluid through a device we call the super sampler that is and it's going to enable us to perhaps filter out any microbial inhabitants of the vent fluid. The key thing we've got to get right at these sites is get the temperature range at which we're sampling.
Just let it go. And then you can just come, boom, there you go. See?
Nice.
Finds itself.
Self-aligned.
Yeah,
Nice
that's why I
Jess.
cut it out all like that.
Way
So
to
you just
go.
let it go and it just drops in.
Nice. All right.
Yeah. I'm just gonna check on where that nozzle ended up there.
Yeah.
Yeah, that should be fine. Good.
Roger.
So, super filters to start.
Yes.
We're leaving the arm out.
Yeah, I think whatever you guys need to do to get the filters ready. That's our next task right here.
So we want to be getting our wand, the main wand ready.
Yeah,
Is that right?
super inlet.
All right. And do you guys need to purge it up in the water column or anything, or is it all good?
No, I don't think so.
Roger.
So let's try and get it out, and then if we need to place it down on the porch and re-grab or whatever we need to do there.
Okay.
Can I get power? The super and
Yep.
Prevco?
Coming up. Power on super, power on Prevco.
Thank you.
Hold on.
Tilt down a little bit.
So Amy, to confirm, we're going to do S1, S2, and S3 for the 3 filters?
Yes, that's correct.
And for your event name, do you want it to be like MC site or something like that? Or do you have a preferred
Actually, I
event
think
name?
I'm gonna name this Mount Edwards.
So just so
Mount
you,
after,
Edwards.
yeah,
before you start,
after
Sounds good.
Katrina Edwards.
you're gonna. It's
Okay.
gonna be hard to get it
She's
out.
a famous
um,
oceanographer. Is no longer
'cause
with
you're gonna
us,
be
but
pushing up against the worms a little bit to keep it low.
yeah,
So
I'm
if
gonna
it's
name it in her honor.
Okay, cool.
if it's too hard, we may need to come off and get it out and then set back down again.
Okay.
And I think we're also dropping a marker, right?
that's a good
so
point.
After all of this, we will.
so
yes.
it will be marker A2 as well, or
Bubble out.
Yeah, so after we do the sampling, we we'll take a rock and then leave a marker. Or leave a marker and then take a rock.
So your arm, look at bubble for a sec. So you're close your
Yeah.
arm.
So
to
for
the
your
tube.
for your sample name, just to be clear for the
So
next
hop off
5
then.
samples.
I think we should hop off. Are you guys okay if we come off and then get the wand out and then sit back down again?
If you're
yeah.
confident that you could come back to the same, yeah?
Yep, I'm confident. Because I can. Those worms are pretty clear.
Yeah,
And they're just to the left of 3.
great indicator.
So otherwise we might risk hitting the experiments and then wiping out a bunch of worms right now.
Yeah, no, that makes sense.
Okay, do you want me to do it?
Sure.
We picked a good site that has some good visual markers, aside from the experiments.
So just to answer one quick question on Nautilus Live, you heard Sean mentioning a lot of the temperature measurements, and some of you are wondering why that doesn't show on the Nautilus Live website. Those temperature measurements are actually taken from a device on the top side of the front of Hercules.
That's
And
carrying it broad, isn't it?
And that probe is well out of the flows that we're measuring. Those temperatures you heard Sean reading off were essentially that was the temperature measured at the very tip of the probe as it was going into the vent flow.
Alright. I'm just gonna turn the bubble cam. Sorry.
Oh, okay, sorry.
No, you're good.
You can get it if you want.
The reason why we're backing off now is that the manipulator
Just so you
arm
see what I'm hung up on.
Yeah.
just has the end of a long wand that's stowed away down there inside the front porch of Hercules, and you have to sort of draw it out bit by bit. And we didn't want to damage the experiments or the tube worm colonies that we were up close to.
Let's rotate a little bit, probably.
Yeah.
Rotation is your friend.
So what you can see there that makes the wand is a blue 3D printed housing that encloses the water inlet tube. Also
my
in
grip's not very good.
Yeah, so get it up on the porch.
Also in that housing are a couple of temperature probes, so we can measure the temperature of the water as it goes into that super sampling device. That hose then flows through the body of Hercules to the super sampler itself, which is a rotating cassette sampler with different filters mounted. And we're going to pump water through there for anything up to a couple of minutes or longer.
Alright. Then we're just gonna pull it back out.
Why don't you, like you don't have a very good grip at all.
No, yeah. I was
So-
gonna just let it go.
Yeah, so why don't you're not quite stuck on the magnet right now. So why don't you get it up and let go of it.
Roger.
And then what our team of microbiologists on board are going to analyze
This
once the
was
ROVs are
nozzle
recovered.
Yeah, it's just stuck on the front of the...
Are those filters containing any particulates that were in the vent fluid, hopefully among them.
Keep bubbling up, there you go. And then pitch.
Hopefully among them colonies of bacteria and archaea.
Yeah.
Yeah, let it go. There. See if you can get a little better grip. I mean, we can also just leave it because Trevor's, they're coming in right now. So
Alright.
you can just let him get his own grip on it too. But he's got it out enough that I think we can land and then still have it wand available.
Roger.
Yeah, if you guys can help him get repositioned.
Oh yeah, I'm gonna get them all lined
perfect.
up here.
Thank you.
Mm-hmm. Josh, are we just repositioning?
You betcha.
Okay.
Would it help to punch in on Argus a little bit?
No.
No.
Hey,
How's it going?
I'm just gonna get you settled into your spot again.
So just for those of you watching on Nautilus Live, over the next 10 minutes or so, we're going to be conducting a watch change as the noon to 4 watch comes in. What this means is
Nicole,
Yes.
what's our depth?
Our current depth, 2707. We're at 3 meters altitude, 2710. Yeah.
710. Okay. Thank you.
But that's not the exact site that we'll be sitting on, so you might want to wait until we just
Oh,
get
okay.
the ROV stable again, and then should be about 2707, which is what you guys were at yesterday.
Okay.
So you guys are going to notice on Nautilus Live some of the comms switch in and switch out as the various team members drop out of the conversation just to bring their counterpart who's coming in up to speed on our operations. So that might disrupt the flow a little bit for the next 10 minutes or so, but then they'll settle down and continue with the super sampler measurement.
Everything's super.
There you are.
Hi, team.
Hi.
Everyone, let's science today.
Okay, right. Has everybody settled in from the 12 till 4 watch now?
More or
We're
less.
just completing
so.
our handover. Okay, just to do a quick introduction. So obviously this is a 12 till 4, and we're going to continue with the super sampling in a minute, and I'm going to hand over to Amy. But just to let everyone know, obviously we've got our Nautilus Live followers that are following us online, and also we have colleagues and scientists at the ISC who are also following. So we're going to keep the SPL as clear as possible to allow Amy to communicate with them so that we can make sure we can log everything as clear as possible to make sure that all of those communications are getting back to the ISC clearly today. Okay?
Okay.
Okay.
Right, I
Sounds
think
like
we're
a
just
plan.
waiting for our data logger to get a headset on, and I think we'll be ready to go. So bear with me 2 minutes.
Okay, and it was my understanding that we're doing the super intake wand with the electrodes and the wand and everything, the temp probe and everything,
Yes.
into the tube worm area. Is that right?
Yes.
Great. Do you want me to wait for data before I get started on that?
I'm good to go.
This yours? It's unmarked. That's Jess's. It says Jess on it right there.
Yep.
Jess, if you're listening, your mug's still up here.
A marked, unmarked mug.
Other
It
Jess.
is marked.
Okay, so I think Aubrey's all ready to go down here, back here on data logger. So Amy and Trevor, we're happy to hand over to you guys to start directing.
Do you guys know the exact spot that we're going to be sampling?
Me?
Did they tell you?
I think I know.
Okay.
We'll get really close and then we'll go from there.
Okay.
Does that sound okay?
Yeah.
Can I please get bubble on the front porch?
Yep.
And maybe on the back of the wand here.
Is that okay?
Yeah, that's good there. Thanks. Right behind the bar. Classic. All right.
Could I also get the flow meter?
Not quite yet,
Okay.
but we will set that up before we're
All right.
flowing. Okay, you can take bubble. Have you done the bubble gauges?
Currently doing that.
Okay. All right, there we go.
Okay. It's okay. It's fine.
Let me think about this. How do I want to do this? That's not bad there, actually.
You want to zoom?
Soonly. Okay, I'm ready for a zoom.
Coming in.
Good there for now. So my understanding was that we're going to go kind of near the dark zone right behind the blue one now, or over there. Is that right? Or
Yeah.
maybe on the
I
back
think-
of this guy too?
Yeah, I think Jess was behind that guy.
Okay, so as soon as these gauge readings are done, we'll take the bubble cam
Okay.
and put it out front. So Cassie will just be a minute doing that, and then we'll get our other perspective. Can you come wide, please? And do you want this to be pretty vertical like this? Is that the desired way?
Whichever way we can get it so the temperature is stable, because that seems like the most difficult part.
Roger, yeah. All right. This one done?
Yep.
Can I have that on the tube worms, please? And you can zoom in on that too. That's good there, Jess. So it's going to be down right near the tip of the wand. Up and left. A little more left. I think we're in the right spot. Yeah, good there. Thanks.
Okay, temperature 7.8. 8, 9. We're looking for about 30 to 35 degrees stability. Okay.
Okay, how's the latency on that? Are we going to have to wait a little bit to see it rise?
It seems to be pretty stable right now, around 8, 9, or 10.
Okay, so I'll kind of go into it a bit more.
Okay. Just try not to go too far so that I don't suck up anything.
Roger that. Jess, you got any more zoom?
It's about
I
11.
do. Say when.
That's good there. Can't really tell if the tip's in the stuff.
It's still around 10 degrees.
Amy, let me know when you've begun actually filtering things.
Okay, do you want me to try moving right a little bit then?
Maybe left.
Maybe left.
Yeah.
Okay.
Where that little orange patch is.
Oh, uh-oh. Come wide, please.
Now you're at 30 degrees.
Oh, okay. Are you happy there? Oops. Oop. Ay.
It's about 25 now. 28, 31, 34, 35, 35, 36, 35. That looks pretty good. It's stable at 36.
Okay. I'm stable here. Is that too hot?
I think 40 is okay.
I can pull
Now
it just slightly out if you'd like.
Ooh, don't do that.
Like a half-inch up, if that would be better.
Yeah, because it's up to 47 now.
Okay, come out just a little bit there, Jess. Good there.
45. 45.
Let's try that.
42. Still about 44.
Okay, higher up.
Yeah. It's 41, 37, 38. That looks good right there.
Okay. Hold it there for a minute, and then if you're happy, then we'll move the bubble cam over to the flow meter. You know where that is?
Yeah.
Go ahead, yeah.
Yeah, it's steady at between 36 and 39. Okay, let me take down a couple of things here.
Data nav, do you want me to log this in HiPAC as well, or we're a little offset from the target from yesterday?
So once we take the sample, I'll have you drop a sample number,
Okay.
sample target.
Okay. Oh, it's going back up now. I'm just going to keep watching it. It's about 45 right now.
It changes pretty crazy, doesn't it?
Yeah, we spent a lot of time trying to get a stable reading. It's at 40.
So you can see, if you look real closely, the vehicle is very slightly moving back and forth, and that might be the cause of this.
Okay.
Yes.
Is that something that
think
we
it's
can
okay.
change,
Or
or is that not worth it? I mean, we're gonna be pumping for like 15 minutes, so
Right. It's not going to get worse.
If we're not that stable, then that might impact the sampling.
We
I
are
don't know if
very
that-
stable, but there's always the possibility we could get more stable. There's also the possibility we get less
That's Amy's
stable.
call. It's just something I was pointing out. That's all.
Yeah.
I mean, it's within 5 degrees is the swing, so I think that that's okay.
I wouldn't expect with the amount it's moving right now for it to move far, but I would expect it to move back and forth, say a half inch, a quarter inch at the end
Okay.
effector there.
Depending which way you move, it could be 122 degrees difference.
That's pretty significant.
Yeah.
We did see that with the IGTs, that
Yeah.
not moving much gave you a very, very different temperature
Yeah.
profile.
Absolutely,
but
yeah.
But you expect at these point sources, you still have like temperature excursions just in that little space within 5 degrees. or
Yeah,
so.
it's-
That's what you're
You're sort of steady around 43, 44 degrees right now.
Yeah.
Really steady.
What was the temperature that we took? 38?
38,
IGTs?
about 38 was the max we saw at the IGTs,
Okay.
I believe, and then the second one was somewhere around 32.
Okay. Oh yeah, that's a lot lower. Could you try to just bring it out or up? Maybe
Absolutely,
just a tiny bit, yeah.
I mean, the other thing is if it's 46 degrees there, just 2 centimeters down, it's probably well into the 60s. So
Yeah.
I'm not sure what we're sampling. We're just diluting the signal with seawater to cool it.
Yeah.
The microbes are still going to be seeing 60-degree water.
Okay, we're pretty stable at 32. 30. 29, 28, 26. It's going back down. Back at 25, 26. So let's move it just a little bit more down back in there. And then,
It's too cold now?
yeah, a little bit.
Okay. Let's see what we got here. Okay, let's give that a go.
Okay. 22. Seems like that's even colder.
You see that's even colder?
Yeah,
Interesting. Okay.
I know.
I'm going to move it again.
Yeah.
'Kay.
Back to the left a little bit, maybe. It seemed like that little patch of reddish yellow was a pretty good area.
Kind of here.
Yeah.
Here-esque.
Maybe even up a little bit more.
Up.
Yeah. Do you see the reddish yellow patch to the left of the wand?
Oh, like 3 inches, 4 inches?
Yeah.
Oh, sure. I can go over there.
We're at 17. 15.
Of course, right now the flow is going in such a way that I can't see it.
I know. Okay, we're back in the 20s.
It really is got that Van Gogh effect.
Yeah, it's really cool.
It's really beautiful, but it's not very helpful.
Or at least we know it's hot.
Oh, you think that's too low?
Has it dropped right off again, Amy? Or is it,
What?
did it drop right off again? Or
It's steady around 20. And
So I
I
can-
really want
I can push it
30,
down a bit
but
more.
you want
Okay.
30?
Are we going to try down?
Sure.
Yeah, you just got to be careful. We're not going to go so low that you're going to suck stuff up.
Whoop. Hey, yep.
25. 23. Can you move it just to the right and down a little bit, like 1 inch to the right and 1 inch down?
Roger.
Uh, 16. It was much hotter here earlier.
Could these little seep vent things be transient?
I suppose.
Showing a very
Now it's 26.
showing a very different temperature than you are.
Are they both in the same location?
I think so.
So I'm around 27 to 30. And Sean's temperature, he also has a probe attached.
Yep.
And his is at 5.
5.
Oh, wow.
Oh.
Okay.
Wild.
Is Sean's
And then they
a little
both
up?
level on the base of the
Yeah.
super, so they're both experiencing, I mean, if we assume the nozzle is parallel to the flow, I mean, one's not up here and one's, because I mean a matter of centimeters from the tip of the
Yeah.
probe would make that difference in temperature.
Yeah.
But
Yeah, we're back down to like 15.
Yeah, I think we need to poke around a little bit more to get a little bit higher, a little bit more stable temperature. It's going to be, we even noted during the IGT sampling, we noticed a very wide temperature swings, and I'm guessing it has to do with just the diffuse nature of the vent, just wafting back and forth. As the vehicle's moving, the flow's moving as well, so.
Okay, well, where would you like me to poke?
Just keep poking around in that same area, I think. Try over to the left a little bit, over to the right a little bit, and see if we can get a good 30 degrees.
I got a good feeling about right here.
I was
Yeah,
going to
right?
say, my
Oh,
gut would
that's too
say
far.
where those
Upper
worm
right.
plumes are would be hotter than where just the snails are. So I think, yeah, probably where Trevor is to the right in that area there would be a little bit, would have hotter water coming from.
It's at 14 degrees.
Oh, instinct is wrong.
12,
It's a pretty quick update, so
10.
The biota are not good proxies, I guess.
All right. 7.
I don't know why I'm showing, I'm showing about 11, 14 degrees right now.
Yeah, and I'm at 9.
What about a little farther away from?
So it's perhaps they're not as co-located as we thought they were, because we were seeing similar temperatures before, but now they're slightly different. Either that or we're not actually in a good concentrated area of flow. We're getting closer now, so I'm at 10 and you're at 14.
Yeah.
Yeah, we're still around 14, and we're looking for close to 30, so.
okay.
So is there a little bit of vertical offset between the two temperature probes?
Yeah, you can. It looks like there might be looks like the titanium one's set back a little bit from the tip of the nozzle.
Mm-hmm.
Okay.
But they still should give similar if we're in a good area of flow,
Right.
similar temperatures. I'm showing about 22 at this point.
I'm 16.
Inverted.
Now I'm 23. 24.
Your thermocouple is really at the tip where you're going to be
26.
sampling, so I would go off that one since I just saw in the video that they're not exactly co-located at the end.
Yeah.
Mm-hmm.
It's crazy that such a small difference can make a huge difference.
30, 33, 34, 35, 36.
Get there.
Promising, 38, 39, 40.
No!
42.
Rats.
Well, we're at least
Overshot.
in the right area that we're getting
Yeah,
higher
you were
temperatures.
very
That we can work
close.
with. We
Great.
can reduce temperature. We can't increase it.
Yep. So we're about 40, 42.
42?
Mm-hmm.
Okay.
Amy, how fast
Okay.
is the super pump?
Actually, hold
A
on.
liter per minute.
All right, so that's going to pump at a decent clip.
Yeah.
So the temperature is, you're going to get seawater entrainment when when you start pumping. So a little bit higher temperature might actually be okay to when it gets diluted when you pump.
Yeah.
So just keep that in mind as we sample, I
Okay.
think.
So we're about 31, 32. Okay, I'm gonna go for it. Let's see. Let me log my time.
You ready, Aubrey?
I'm going to try and bump the camera here.
Looks like right now the temperature is pretty stable at about 31, 32 degrees.
Yeah. Okay.
If you can just shout out, Amy, when you start the process.
Are you locked in?
I'm
if
locked
I
in,
start
yeah.
sampling? Okay.
Yeah. Stable, yes.
Okay.
Yeah, I'm ready.
I'm going to be moving to home. It will take just about a minute before I start sampling.
Great, thanks.
So I'm showing about 15 degrees on my thermocouple.
I'm down to about 22.
Are you good with that?
It's stable.
Okay.
Okay, I'm at port position 1. I'm going to filter slow. Start time.
Yeah, temperature's dropping off a little bit.
Yeah.
And Amy, when you say filter slow, do you have a speed for that?
It's about 1.3 liters per minute. So I should be filtering for about 15 minutes or so to get 15 liters.
Perfect, thank you.
So it has started pumping already?
Yeah.
Okay, and the temperature on
I
the
see the
super
flow
is about
meter moving.
19 degrees C.
Yeah.
Hi Amy, did you flush the bag as well or filter?
I'm not filling a bag right now. We're just doing filters.
Got it, thanks.
Sure.
Temperature on the super is about 20 degrees. Temperature on the IGT electronics are about 20 degrees, so that looks like the temperature is very close now. Super is now at about 24 degrees. Looks like it's pretty stable at around 22, 24 degrees.
Yeah, temperature's staying stable.
Yes, about 22 to 24 degrees, it looks like from what I can see on your screen. Just giving updates to those that may be listening in and wondering what's going on as the wand is just sitting there.
That's
and
great.
They don't have the luxury of a computer screen in front of them.
And also the paddle wheel as well. Are you guys happy with the flow of that? That's quite consistent at the moment.
Looks good. Yeah.
Good.
But yeah, both Super Wand and IGT electronics are both at about 22 degrees now, so that's
good news.
Indicating that we might have a pretty decent flow there, enough to get good stuff into the wand.
Is this Sean?
Yes, it is.
Yeah, hi Sean, it's Susan. Haycock Academy had a question about the instruments and what they're made of to withstand the temperature and the pressure.
I'm sorry, could you repeat that? It was a little...
What, the instruments, what are they made of to withstand the temperature and the pressure?
Oh, I'm not sure about the Super, but I can tell you that the IGTs, or isobaric gas tights, are made of titanium. It's a titanium alloy, which gives it very high strength, and it's quite lightweight. We've designed them to go down to not full, but 4,500 meters. So we test them at about 10,200 PSI, both internally and externally, to make sure that they can withstand the pressures that we're going to subject them to. However, that 10,200 is it's about 1.5, I think, times what they're rated to. So that we build in a safety factor. We test them at much higher pressure than we would actually use them at.
Mm-hmm.
On the IGTs also, the electronics are at atmospheric pressure inside a pressure housing.
Mm-hmm.
So we also test those as well. The snorkel and the thermocouple on the IGTs are all made of titanium as well. And the reason for that is because we measure metals. and stainless steel would contaminate the samples, and the titanium does much better with much higher temperatures and acidities than the stainless steel would. Some of the vent fluids that we sample are at about pH 2, so Coca-Cola acidity, and they're quite corrosive, as well as being 300 to 400 degrees C. Not 400 here, only 300 degrees here, but.
Thank you. And is that cover on the temperature probe, was that 3D printed?
That's super related, but I believe Eric said that that was 3D printed, but you'd have to ask Eric that.
Okay.
Looks like temperature is still holding steady at about 24, 25 degrees on the super and about 30, 31 on the IGT Electronics one temperature probe.
Hey Jess, you can bring a closer shot to Herc if you'd like. Sure. Look at that robot.
Super one, the temperature is very, very stable.
Yeah, that's good news.
I would love like a wall painting of this Herc Zeus view without the science gear in it, but just like Van Gogh style.
A control print.
It was the artist that came out a couple like 2 years ago.
Yeah.
Got to commission her.
Yeah.
I think we would argue that the science gear makes it.
I mean,
You want just a full wall mural, wall to wall, end to end?
just science gear.
Well, it adds that extra bit of visual interest.
With that Van Gogh flair. Can we get a
Super
quick
sample is still around 22, 23 degrees.
Thanks, Sean. Took the words out of my mouth.
Looking good.
You took the words out of my mouth. I was just about to ask. Can I get a quick update?
Yeah,
That's great. Thank you. Love
IGT
that.
thermocouple is about 33 degrees, so still holding steady
Yep.
during the pumping, which is good news.
We're about halfway through this filter.
Brilliant.
You guys said the super wand is around 23 and the IGT is 33?
Right now, super wand is 25 degrees,
Okay.
and IGT thermocouple is 34.
Okay.
They've tended to waft back and forth between being close and being further away, and I think that just might have to do with the currents pushing the hydrothermal fluids around. Diffuse fluids are very wafty by their nature and very subjective.
Wafty.
Wafty, yes, the technical term.
I like that word.
That's a good term.
They're easily influenced by bottom currents and thrusters, so they kind of waft back and forth, swaying in the wind, if you will. So super wand now is at about 27 degrees, and IGT electronics are at, IGT thermocouple is at 43.
What are we looking at in bubble cam right now?
Bubble cam, we're looking at
I don't know if you want to pick that up, Amy. I think that's the flow meter.
That's a 13. In bubble cam, we're looking at the flow meter.
Is that the flow meter?
Yeah, the flow meter.
Super wand still about 25 degrees.
Super.
IGT thermocouple is 36 degrees.
Try now.
Hey Jess,
Hey, Cassidy. So what are we seeing in bubble cam?
we're looking at the flow meter.
Oh
It's a
yeah, do you want
flow
to explain,
meter.
Amy, what the flow meter is and how that works? Is it just basically, I think it gives us an indication, doesn't it, of how the flow rate is changing. So
Yes.
if the paddle stops, we know we're not actually bringing water into the super
That's
sampler.
correct.
So it's just a visual way of confirming that we're actually keeping a steady
Yes.
flow rate. Is that correct?
Yes,
That's
and
my
it's also
understanding.
counting.
And it counts
so
as
it
well.
will tell us, and when I do some calculations in a spreadsheet, it will tell us. In this program for the super, you have a flow count when you start, and then a flow count when you end. And if I enter that into the spreadsheet and do some magical math, it
Yeah.
tells me how many liters I was able to filter.
Perfect. So it'll give you a final volume of what has actually gone across your filter, so you can start working out densities and things.
That's correct.
Super one now is about 22, 23 degrees, and IGT electronics, well, thermocouple about 31.
The guy poked his head out again.
Copying. Yeah.
Super temperature is still 22 degrees. IGT thermocouple's at 26.
Oh. It might be important.
We have about 5 more minutes for this filter. We're doing 3 filters today at this site. For each of the low temperature vent sites, we're collecting 3 filters worth of microorganisms. We have different procedures to extract DNA or RNA or metabolites, and that will give us a good picture of the entire community.
What's the diameter? What's the pore size of the filters, Amy?
0.2 microns.
0.22 micron?
Yeah.
So that should filter out all bacteria,
Yeah, there might be
eukaryotes,
some
ciliates,
that go
anything that's in
through.
there?
Yeah, there might be some that get through, but mostly that's a good size to capture everything.
And do you utilize the whole filter as one sample, or do you split that into subsamples? Do you like divide that up into separate samples for replication?
For Loihi, I cut the filters and I only used a small portion of it, because these filters are larger than the ones that Julie's used before, and so we didn't know if we needed to use that whole filter. And it's always nice to have some backup, so we just kind of keep it in the freezer and only use what we need. And if we get enough, then we'll just save the rest.
Understood. Yeah. So you have that backup if you need it.
Yes. Yeah.
Super one now is at 23, 24, 25 degrees, sort of stable in that range. IGT thermocouple's at 41 currently. I wouldn't expect that much of almost a 20-degree difference over that short distance, but
yeah,
it's appears to be real.
If you have a moment, Amy, to address, what are some of the unique features of the enzymes that can withstand these high temperatures within the bacteria?
I guess they're resistant to being denatured at high temperatures. So we actually use a lot of enzymes that are from thermophilic organisms in the lab. In particular, there's one called TAC polymerase, which was isolated from an organism called Thermus aquaticus. And that enzyme is really key into a lot of our sequencing ability and our ability in general to know the genetic information from these organisms and being able to sequence them and determine what their metabolic capabilities are. Mm-hmm. It's really important because we can't always get these guys in culture. And so this is really the best way that we can investigate the entire community and try to understand how complex it is and the ecology of these organisms and how they live together. And yeah, so it's a really important discovery when and was actually like a giant leap forward in biology and molecular biology when that enzyme started being used for polymerase chain reaction, which is what we use. Yeah, so that's the main one that I always think about when I think about high temperature enzymes.
Super wand is at 26, 25, 27 degrees in that area. IGT thermocouples is about 46. Super wand went up to 28. The higher temperatures I've seen.
I've seen 30 a couple
Have
times.
you seen 30?
Yeah.
I've been just sort of casually looking to sort of keep updated on what the temperature's looking like over the course of the filtering.
If we could keep an eye on what the max temperature has been throughout this process too, they're interested in that on shore.
Yes, it's, so far I've only seen 30.
IGT electronics say 61.
Oh.
Now whether or not that's actually made it in the end of the super wand or not is unclear. I think the IGT electronic thermocouple, we may want to shift a little bit towards the end where the super wand is. Maybe we can get them a little bit better co-located when it comes up.
Okay. Okay, so I'm going to stop this filter.
So just to confirm that was
Filter
end of
stop.
sampling?
Let me enter my info.
Are we going to be doing another filter in the same spot?
Yes.
Okay.
3 filters total.
Roger.
So just to confirm, that was a flow rate of 1.3 liters per minute and a final volume of 15 liters?
Yes.
Sean, at some point, could you please explain IGT to the audience there?
Sure. An IGT is called an isobaric gas tight. IGT is for short. Isobaric meaning same pressure. So what's unique about those is that we can take a fluid sample at the, not the bottom of the ocean, but at depth at 2,700 meters, and we can bring it up to the surface, maintaining that 270 bar of pressure. And because of that, it keeps any of the gases that are dissolved in the fluid in solution in one phase. It's kind of like as simplistic as bringing up a can of Coke, and when you open the can of Coke, you want to get all the gas that you just released out of it in a controlled manner. It's an overly simplified version of it, but that's essentially what we're doing.
Thank you.
But the super sampler is something entirely different. With the super sampler, they're more interested in particles and in the filters, and then water in the bottles, in the bags that will be sampled later.
Okay, appreciate it.
Okay, just to confirm, we have started the second filter, so the
Yes,
data logger
that's
has logged
correct.
that. Yeah,
Yes.
good. Yeah, we were on that. So in terms of switching the filters, is it just something you're able to do remotely from your laptop? And obviously we're not changing the position of the snorkel or anything. It's all done at the back where those filters are, so it's just changing the channels, as it were.
Yes, that's correct.
To switch the water flow over a separate chamber.
Mm-hmm.
So start of the super sampling now, we're at 24 degrees.
Yeah.
And IGT thermocouple's at 40. Super one thermocouples hovering around 20, 21 degrees. T thermocouples at 49. Difference.
keeping an eye on the paddle